Good afternoon, distinguished guests, colleagues, and friends. It is my tremendous honor to welcome you to the 2025 Dr. Stanley Ho Memorial Lecture. I'm Professor Maggie Wan from the Chinese University of Hong Kong, and also the member of External Advisory Board of Center for Personalised Medicine, University of Oxford. And I'll be your MC today. We gather to honor the legacy of Dr. Stanley Ho, whose visionary philanthropy continues to inspire innovations in medical science. And today's lecture promises to be a milestone in our ongoing pursuit of knowledge. Before we begin, allow me to acknowledge our distinguished guests today, whose presence graces this occasion. The Vice Chancellor of the Chinese University of Hong Kong and Scientific Director of Center for Novostics, Professor Dennis Lo. Ms. Helen King, Principal of St. Anne's College, University of Oxford. Professor Anna Lucassen, Director of Center for Personalised Medicine, University of Oxford. Mr. Patrick Huynh, Chairman of Dr. Stanley Ho Medical Development Foundation. Mr. Ian Huynh, Vice Chairman of Dr. Stanley Ho Medical Development Foundation. And our distinguished lecturer today, Professor Sir Peter Donnelly. To all guests, colleagues, and friends, welcome. To commence today's program, I would like to first invite Professor Dennis Lo to deliver opening remarks. Professor Lo is pioneering the genomic research by liquid biopsy technology. Professor Lo, the podium is yours.
Distinguished guests, friends, esteemed colleagues, ladies and gentlemen, good afternoon. It's my great honor and pleasure on behalf of the Chinese University of Hong Kong and the Center for Novostics to welcome you all to the 2025 Dr. Stanley Ho Memorial Lecture. This lecture series pays tribute to the visionary legacy of Dr. Stanley Ho, whose extraordinary philanthropy has left a profound and enduring impact on education, medicine, and public health. At the Chinese University of Hong Kong, we're deeply appreciative of his generous support, which led to the establishment of 3 research centers: the Stanley Ho Center for Emerging Infectious Disease, the Stanley Ho Big Data Decision Analytics Research Center, and the Brain and Mind Institute. In addition, the endowed Professor Stanley Ho Professorship in Respiratory Medicine and Cognitive Neuroscience further exemplify his lasting legacy. And together, these contributions have empowered researchers to combat infections, addressing public health challenges through data-driven solutions, and further neurological research to enhance development and quality of life, and a mission that remains critically important in today's world. It's especially meaningful that this annual lecture is being held in Hong Kong at CHK for the first time since its inception in 2021. And this year also marks the 20th anniversary of the Dr. Stanley Ho Medical Development Foundation, a milestone reflecting 2 decades of unwavering dedication to improving human health and well-being. As we gather here today, we not only honor Dr. Ho's legacy, but also reaffirm our collective commitment to advancing global medical innovation through research, collaboration, and education. And today, we are privileged to welcome an exceptional speaker, Sir Peter Donnelly, whose groundbreaking contribution to statistics, genetics, and precision medicine has exemplified the spirit of innovation and excellence that this lecture series seeks to inspire. I myself also work in the area of genetic diagnostics, and I'm certainly a fan of Sir Peter's exceptional work. Also, I'd like to take this opportunity to express my heartfelt gratitude to Mr. Patrick Huhn for his stewardship of the Dr. Stanley Ho Medical Development Foundation and for his support to CHK over the years. My sincere thanks also go to the University of Oxford, St. Anne's College, and the Center for Personalised Medicine for their key roles in establishing this distinguished lecture series. And I'm especially pleased to see Ms. Helen King, the principal of St. Anne's College, join us today at CHK. I also had the pleasure of serving on the external advisory board of the Center for Personalised Medicine for a number of years. I'm very happy to see that the current director of the center, Professor Annika Lucassen, is also here. Actually, Annika and I had collaborated probably 2 or 3 decades ago, right, in the area of diabetic genetics. So I really treasure this opportunity to catch up with you. So may today's lecture inspire all of us as we continue to work together towards a healthier and brighter future. So thank you.
Thank you, Professor Lo. Next, we welcome Ms. Helen King, Principal of St. Anne's College, University of Oxford. Helen's leadership bridges innovations and cross-discipline collaborations. Helen, please share with us.
Thank you. I'm very honoured to be here representing St. Anne's College, the University of Oxford, and the Centre of Personalised Medicine to express our gratitude to Professor Dennis Lo and to the Chinese University of Hong Kong for hosting this, the 5th Dr. Stanley Ho Memorial Lecture, which was instituted by the Centre of Personalised Medicine and up to this date has been held in the University of Oxford. But those lectures are available on the Centre of Personalised Medicine website, including with translations. So please do look back at the eminent lectures that have happened in the past. The Centre of Personalised Medicine is a very special partnership between the University of Oxford Centre for Human Genetics and St. Anne's College, which has been generously funded since its inception 12 years ago by the Dr. Stanley Ho Medical Development Foundation. The foundation is 20 years old, and we are privileged and honoured to be able to celebrate with you the 20 years of achievements and the impact that it has had, not just here within the region, but across the globe. The Centre of Personalised Medicine was the brainchild of 3 visionary men. My predecessor, Tim Gardam, who was principal of St. Anne's College, our speaker tonight, Professor Sir Peter Donnelly, who at the time was a fellow at St. Anne's, but also, and very importantly, Mr. Patrick Huhn, the chair of the Dr. Stanley Ho Medical Development Foundation. They were visionary in seeing the importance of personalised medicine and the opportunities that could be created to bring together researchers, clinicians, students, patients, to share understanding and knowledge, not just in Oxford, in the UK, but also in Hong Kong and Macau. And by sharing that understanding and insight in personalised medicine, how we can accelerate its application and its effectiveness in healthcare and improving the quality and length of life for individuals and populations across the globe. So this isn't just an exciting event and an opportunity to celebrate what has already been achieved, but it marks the fact that we are in exciting times where there is much potential for all working in the field. We are so deeply grateful to the Dr. Stanley Ho Medical Development Foundation, into the philanthropy of Dr. Stanley Ho, who we remember with great respect, to Katie Poon and the team at the Medical Development Foundation, of course to Mr. Huhn and to all his family for the support he so kindly gives the Centre of Personalised Medicine, St. Anne's College and the University of Oxford, and to our many advisors and collaborators, including Professor Dennis Lowe and indeed Professor Joseph Sung, who are previous members of the External Advisory Board. And we're delighted and honoured that our new member of the External Advisory Board is joining us now. As a result of all your work and ongoing work, the connections we're creating, both for now and for the future, the world will become a more knowledgeable and healthier place. We're hugely grateful for that ongoing support and honoured to be here today. Thank you.
Thank you, Helen, for highlighting the power of collaborations across all disciplines. As we celebrate the 20th anniversary of Dr. Stanley Ho Medical Development Foundation, we are privileged to invite Mr. Ian Huynh to share with us the legacy of the foundation. Mr. Huynh will also introduce our distinguished lecturer today. Mr. Huynh, the stage is yours.
I've been given the onerous task of introducing Sir Peter, so I need my cheat sheet because I need to tell everyone at least a bit of all the accomplishments he's had, but I don't have enough time, so it's just going to be the tip of the iceberg. So thank you very much, distinguished guests, Professor Lowe, Mr. Huynh, Ms. King, Sir Peter. I am Ian Huynh. I am the Vice Chairman of the Dr. Stanley Ho Medical Development Foundation, and welcome to our 2025 lecture. The foundation really is the embodiment of Dr. Ho's love for Macau, and above all, the people in Macau. So our motto has always been, our central goal has always been for the betterment of the health of the Macanese by having international collaboration for cutting-edge technologies for the improvement of healthcare. And I think today the event completely embodies that principle of our foundation. We have, thank you to the generosity of Vice Chancellor. We are hosting the Center for Personalised Medicine lecture here in CUHK. We have a distinguished professor from Oxford, Sir Peter. And so this is really an event where we're putting Macau, Hong Kong, and Oxford together, talking about a cutting-edge technology to help lives around the world. And for us, it's about specifically for the lives of the Macanese people. So this really is an international collaboration. And I think, and it suits very well with Sir Peter's background. He's, I would say, an international man of science. So he is originally from Brisbane, Queensland, Australia. He studied at the University of Queensland. This is where the reading comes in. It's going to be a long list, sorry, but I have to do it. So, okay, bear with me. He was formerly the professor of statistical science at the Wellcome Centre for Human Genetics at the University of Oxford. So after Queensland, he earned his doctorate in Oxford as a Rhodes Scholar, and he also held professorships at the Universities of London and Chicago. Returning to Oxford in 1996, he has since been nominated as the Fellow of the Royal Society and of the Academy of Medical Sciences. He's an honorary fellow of the Institute of Actuaries. He's an honorary fellow for St. Anne's College and also Balliol College, Oxford. And last but not least, he's been awarded knighthood in the Queen's Birthday Honours List in 2019. He has an honorary doctorate degree from the University of Melbourne in 2022, and my understanding is that he will be traveling very soon back to his hometown and receive the honor of honorary doctorate from his alma mater, University of Queensland. He'll be speaking on the power and potential of genetics to improve healthcare. I've been given some pros to introduce the topic, but I think you would rather hear from him than from me. So thank you very much, and Sir Peter, it's all yours.
It's a great honor and a great privilege to be asked to give the Dr. Stanley Ho Memorial Lecture. A particular privilege to be able to deliver that lecture in Hong Kong amongst many of the people who have worked so tirelessly to take advantage of the legacy he left. I want to start by acknowledging his visionary generosity, his philanthropy. It's helped in many, many different ways, but it's had a major impact through the Dr. Stanley Ho Medical Development Foundation on our work in Oxford, principally through funding the Centre for Personalised Medicine, which has enabled the collaborations amongst different parts of the university in Oxford, but even more importantly, collaborations between those of us in Oxford and those in this part of the world, Hong Kong and Macau in particular. I also want to take the time to acknowledge and thank Mr. Patrick Huon. Patrick, as you know, is chair of the foundation. He, as Helen absolutely rightly said, was a key part of the vision behind the Centre for Personalised Medicine. He's been a great supporter over many years of the centre. But even more importantly for me, he's become a great friend. And I cherish my friendship with Patrick and with his family, and it's wonderful to be able to spend time with you. So I'm talking today about personalised medicine. Personalised medicine is a subject many of us have been going on about for about 20 or 25 years. It was always just about to happen. I think many of those expectations were too enthusiastic and ahead of their time. I think we now have personalised medicine in some cancer treatments, not least through the technology that Professor Lowe has pioneered and is absolutely world-changing. But across many diseases, we are no closer to personalised medicine now in clinical practice than we were 20 or 25 years ago. But that will change, and I want to tell you the story that has led us to this and some of the technology that is changing it. Until a few years ago, if I had the entire DNA sequence of someone who is healthy and 40 years old, I would have learned something medically actionable in about 1 or 2% of cases. And the reason for that is that historically, genetics has played into clinical medicine through looking at conditions due to single changes in our DNA, so-called mutations, that have usually severe consequences. So if you think of conditions like cystic fibrosis or Huntington's disease or muscular dystrophy, they're examples where genetics, a single change, has a big impact. Thankfully, those conditions are fairly rare, although they're very serious for those who have them. Not only are they individually rare, they're actually quite rare collectively. Furthermore, many of them have their impact early in someone's life. So if you get to age 40 and you haven't had any of the phenotypes or the consequence of these conditions, there's probably not much left to find of that kind in your DNA, which is why we only have the 1 or 2% where knowing the DNA sequence would have been helpful. That has now changed entirely. Now, if I have the DNA information from a healthy 40-year-old, I learn something medically actionable in about 70% of cases. And that change from 1% to 70% is because we can now measure the genetic component of risk for all of the common diseases. Diseases like heart disease, stroke, diabetes, breast cancer, prostate cancer, and many of the other common cancers. I'll say a little bit more about how we do that, but that's the change to all the common diseases that has made a big difference. Yesterday in the UK, the Prime Minister, Sir Keir Starmer, announced the government's 10-year plan for our National Health Service. A key part of that plan was moving from sickness to prevention, to enable the NHS, and it's true of many other health systems, to become sustainable by putting more effort into preventing disease than waiting till people get sick and treating them. Absolutely central to that 10-year plan was the idea of using genetics and genomics to understand risk for individuals. And that's what my story will be about. I'll tell you what has led us to this. I'll talk to you about some of the technology, where we are now, and what I think that can do in the future. As I said, for some diseases, genetics is the whole story. Single change means that someone will get sick. over the last 10 or 15 years, our ability to understand and learn about those genetic causes has grown substantially. We were lucky in Oxford to be one of the pioneers in a collaboration with Illumina, the sequencing technology company, to try out whole genome sequencing in the clinic. And the story here from the Times at the time of the study was about one of our early findings where we were able to provide diagnosis for a young girl who had a serious condition, but until then, neither her parents nor her doctors had known what was causing it. And that area, it's called genomic medicine, has been transformative over the last 15 or 20 years. It's an area which the NHS has played a leading role in and continues to do so to this day. Indeed, another announcement in the NHS long-term plan is to move to screening of all newborns in the UK, exactly to look for these kinds of conditions early enough so that in some cases you could take actions to avoid them. But my story is about the common diseases, not the rare genetic conditions, diseases like heart disease and diabetes and breast and prostate cancer. Genetics has a big impact on those, but we know from many years of genetic studies that it's much more complicated. There's not a single place in our DNA or a single gene that affects our risk for heart disease. It's many, many places. And we know that through a series of studies called genome-wide association studies. We started to be able to do these studies in about 2004 or 2005. Until that time, in spite of 20 years of research, we had maybe a handful of examples where we knew individual places in our DNA which affected someone's risk for one of the common diseases. So what's a genome-wide association study? It's not a very complicated idea. The idea is you take a large number of people who have a particular disease, say heart disease, and a large number of people who don't. And you measure them at many places in their DNA, say 800,000 or 1 million places. And then you compare each one of those places in the sick people with the healthy people. And what you're looking for are places where a genetic variant is more common in one of those groups than the other. Because if you think the other way, if there were a genetic variant that made you more likely to get heart disease, that would be a bit more common when you looked at people with heart disease. That's what these studies did. And the big breakthrough that allowed them was the ability to measure those 800,000 positions in our DNA relatively cheaply. The figure on the right are the findings from one of the early studies. It's something called the Wellcome Trust Case Control Consortium, which I was privileged to lead. What's shown in the picture, although you can't see it, each row is a different disease, and there are dots for each of those 800,000 positions. Most of the dots are coloured either light blue or dark blue, in deference to the colours of Oxford and Boo, Cambridge. But those which stick up in green are regions where there's a very strong signal of a difference in frequency between the sick people and the healthy people, which point to a genetic variant which is associated with susceptibility to the disease. And as we started to do these studies, people started to chart their impact. I'll show you a lot of pictures like this. The long stripy things in the picture are ideograms of our chromosomes. We have 22 pairs of chromosomes, in which we get one from our father, one from our mother, and then women have two X chromosomes, males have a Y chromosome and an X chromosome, 23 chromosomes altogether. And the little lollipops, there's only one on this picture, which is here. They mark a specific place for a particular disease where there was a finding that that position was associated with susceptibility for the disease. It was one of those findings in 2005. There are a few more in 2006. In 2007, there was a major change. Our large study, the Wellcome Trust Case Control Consortium, reported it was responsible for about 20 of those findings, but a number of other studies reported. And for those of us who'd been trying to understand genetics for common diseases in humans, this was an extraordinary time. After 20 years of getting absolutely nowhere, we suddenly had many, many clues to susceptibility to the disease and hence to the diseases themselves. This had an impact. So at the end of each year, Science magazine, Science and Nature are the two premier scientific publications, perhaps rather pompously, Science magazine at the end of each year steps back and says, what was the most important development in all of science during that year? And in 2007, they chose this transformation in our knowledge of the genetic basis of common human diseases. Our own studies attracted attention in the press in the UK, some of which was broader than that. This is from one of the newspapers, The Independent. They were very excited about our findings for many different diseases. Rather interestingly, we shared the front page with a woman called Tracey Emin, who is a well-known, very avant-garde and rather controversial modern artist. So it was an interesting juxtaposition. One of the other newspapers, we shared it with the dizzy heights of England, the England soccer team beating Denmark 2-0 in a World Cup qualifier. So maybe Tracey Emin's art is more appropriate. But the work kept going. We were able to do these studies. They were larger, and so we learnt more as we continued them. That was the position by 2009. We did a lot by 2010. That's the current position. We now know well over 100,000 places in our DNA associated with either diseases or changes in things we measure in people, like blood pressure, cholesterol levels, and so on. It's been absolutely extraordinary. Each of those findings is a potential clue to the biology of the disease, and we and others use them to help find new drug targets for diseases where there is serious unmet need. But they're also critical in being able to predict risk for those diseases. And that's the main focus of the talk today. The key idea there is something called a polygenic risk score. I said there wasn't one gene or two genes involved in susceptibility to heart disease. There are many places in our DNA. So the idea of polygenic just means combine information from many places in our DNA. And it turns out that there are 1 million or more places in our DNA that affect someone's risk for heart disease. There are different 1 million or more that affect a woman's risk for breast cancer, different 1 million that affect a man's risk for prostate cancer, another 1 million or more that affect someone's risk for diabetes. We can now measure those. We've got large enough studies to identify them well. And then we can use sophisticated algorithms that use machine learning and high-powered statistical tools to combine that information to get an overall summary for a person of their genetic risk for heart disease. From the same data, you can also calculate an overall summary of their risk for diabetes. For a man, his risk for prostate cancer. For a woman, her risk for breast cancer. And these scores can have a big impact. So what's shown on the right of the picture here is data from 100,000 men in a very large study called UK Biobank. These men were studied forward in time for more than 10 years. We used our genetic scores to divide the men into 3 groups. And for the statisticians amongst you, the genetic scores were trained on separate data. This is independent test data. So there are 3 groups shown in the picture: those with high genetic risk in red, those with typical or medium genetic risk in green, and those with low genetic risk in blue. And then what the figure plots is that at each age, what proportion of each group have been diagnosed with heart disease. So if you look at the high-risk group, by age 50, something like 15% of men have been diagnosed with heart disease. By age 70, it's around 40%. They're the men with high genetic risk. For the men with low genetic risk, effectively none of them have heart disease by age 50, and even by age 70, it's 2 or 3%. There are massive differences in risk. And another thing which follows from that is that the risk accrues earlier in life. So if you take a typical man in the high-risk group, his risk in his mid-40s is more like that of a typical man when he's in his early 60s. Currently, we ignore this information in the way we think about preventing heart disease, but we can now take account of it, as I'll explain. That's heart disease. Here are similar figures on the left for breast cancer and on the right for prostate cancer. Breast cancer is a little bit complicated because there are a number of genes where individual mutations have a big impact on a woman's risk. The BRCA genes are the two most well-known examples. This is ignoring those genes. This is looking at the cumulative effect of 1 million or more places in a woman's DNA. Again, you can calculate a polygenic risk score, summarize her genetic risk, and women with high genetic risk, almost 30% of those will have been diagnosed with breast cancer by the time they're age 70 or 75, whereas for those with low genetic risk, it's only a few percent. The differences are even more stark for prostate cancer. Men with high genetic risk have something like a 40% risk by age 70. Men with low genetic risk have a 1% or so risk. Massive differences in risk. I'll take you on a diversion and talk about BMI, body mass index. It's a major health challenge in much of the developed north, not so much, to your credit, in this part of the world, but a major focus in countries like the UK and the US and Europe. This is data from a study which followed children from birth, called the ALSPAC study. They were born in 1990 and they've now been followed for more than 30 years. There's genetic information available on those individuals, so we could calculate their genetic risk for obesity or for high BMI. Again, that's the result of the impact of 1 million or more places in our DNA. The first figure here on the left shows the average BMI for 10 different groups of the children. The left-hand point are those with the lowest genetic predisposition for BMI. The next is the next lowest 10%, and so on. And the right-hand point are the kids with the highest 10% of polygenic risk score for BMI. Even at 8 months old, which is what that picture shows, you can start to see a relationship between genetics and the BMI of the kids. By 3.5 years, it's more obvious. By 8 years old, it's really striking. And by 18, it's more obvious still. That's what we call cross-sectional data, looking at averages. The other thing you can do is to take an individual child and trace their BMI over time, and you get a curve. And then we can average those curves for people with high genetic risk. That's the red curve in the picture, low genetic risk, the blue curve, and typical genetic risk, the green curve. And again, what you see is those, consistent with the picture I've just shown you, those with high genetic risk have BMI which is higher, substantially higher than average, and increases so that by their early 20s, on average, those individuals have BMI above 30. And that's the definition, or one of the definitions, of obesity. In contrast, people with low genetic risk have BMI which remains at a healthy level throughout the range where they're measured. Those curves are averages for each individual. Here are some of the individual curves. The red curves, each red curve is one of the children in the study. It's their BMI at different ages. And the only point I want to make here is, although the red and the blue curves I showed you previously, and they're on this line, this graph as well, are averages, what's striking is that almost all of the red curves, the BMI curves for the high genetic risk people, are above those for the low genetic risk. It makes a big difference. This is data on 2 different groups. This is one of them, I'll show you another in a minute, of girls on the left and boys on the right. It's plotted in a slightly different way. So what you see here is, for this group of girls, we show their actual BMI, or the average for the group, as you go from those with very helpful genetics on the left-hand side to unhelpful genetics on the right-hand side. And what you can see is, for that group, as you go from unhelpful to helpful genetics, BMI goes up by about 6 units from 12 to 18. There's now a different group shown on each figure, the pink curve. What's striking is that now, when you go from unhelpful to helpful genetics, BMI goes up by 8 or 9 units. So genetics has a much bigger impact in the pink group than the blue group. What's the difference between the 2 groups? The pink group are the kids with high calorie intake, and the blue group are those with low calorie intake. The same is true if you look at activity levels. Those with high activity levels show less of an impact of genetics on BMI. Those with low activity levels show more. This is in childhood. The same is true. Others have shown it, and we have shown it in UK Biobank in adulthood. So it's a sort of double whammy. If your lifestyle is unhelpful, then genetics has even more of an impact. On the other hand, changing your lifestyle, improving your calorie intake or your exercise regimes, reduces your BMI regardless of your genetics. I talked about childhood. This is adulthood in UK Biobank. Again, 3 groups of people, depending on how old they were when they joined UK Biobank: 40-year-olds, 50-year-olds, and 60-year-olds. Again, each point is 10% of the group. It's the average BMI of 10% of the group, with each point showing an increasing slice of polygenic risk. And what you can see in each of the age groups is those people in the top 10% are on average obese in middle age. I won't bore you with too many data points, but another one I quite like is the following. If you take people who have a healthy weight between age 25 and 35 and ask how likely are they to become obese later in life, those with unhelpful genetics are 10 times more likely to become obese than those with helpful genetics. We all know the advice to exercise each day, to walk as many steps as you can. 10,000 is a number that's often quoted. This is a really interesting study by colleagues in the US who had data following people through time where they measured their activity level, how many steps they took, they measured their BMI, and they knew their genetics. So as you might imagine, if your genetics is unhelpful, then over time your BMI will tend to go up. Other things being equal, if you exercise more, walk more steps, your BMI will tend to go down. So you can ask the question, how many steps do you need to walk a day to balance out your genetics and keep your BMI stable? That's what's shown on this curve. It says that if you're at the 20th percentile, so you've got very helpful genetics, not super helpful, but pretty helpful, you need to walk about 3,500 steps a day to keep your weight stable. If you have unhelpful genetics, you're at the 95th percentile, you need to walk 13,500 steps a day. So there's a difference of 10,000 steps a day people have to walk to keep their weight stable based on their genetics. All of the results I've talked about to date are for people of European ancestries and for the obesity work in European environments. I'll come back a little to the way this story pans out for people of other ancestry groupings. For some diseases, and I'll talk a bit more about cardiovascular disease, it's very natural to combine the genetic component of risk with the risk calculators that doctors already use. They already use a tool. In the UK, it's called QRisk, which combines someone's age, their sex, their blood pressure, their BMI, their cholesterol level, and so on. Genetic risk via PRS, polygenic risk, is independent of that clinical risk, so it's capturing something new. For men between 40 and 55, the genetic component of risk by itself is as important as all of those clinical risk factors. For other diseases, it turns out genetics is almost all the story of risk. And the figure on the right of the slide is for breast cancer. There are a number of known risk factors for breast cancer. They're listed there. Each bar is a measure of how predictive that risk factor is by itself. So on a particular scale, the so-called area under the receiver operating curve, larger numbers mean more predictive. So a woman's BMI, whether she's used hormone replacement therapy, her age when she started her periods, her alcohol consumption, her age at menopause, the impact of things like BRCA mutations, parity is the number of children the woman's had, their height, and their family history. They are all important risk factors for breast cancer, but individually they don't contribute that much. And indeed, the third bar from the top is the combined impact of all of these risk factors. And on this scale, their impact is 0.59. if you just have the polygenic risk score by itself, it's much more predictive than all of the other risk factors combined. That's comparing the blue bar with the one below it. And indeed, if you add in all of the other risk factors to polygenic risk, you improve your prediction, but only by a smidgen, only by a tiny amount. So in fact, if you're predicting risk for breast cancer at a population level, polygenic risk is the key driver of risk. Same is true for prostate cancer and for colon cancer. 11 years ago, I founded with colleagues a company called Genomics Limited to take the advances in our science and get them into the clinic. Separate journey about being an entrepreneur. I'll just say in passing, in my experience, Dennis does these things wonderfully well, but in my experience, it's a much tougher gig than being an academic. But one of the things we've done is to focus a lot on developing polygenic risk scores. And as a consequence, we have the most powerful technology available. This slide, each row in the slide is a disease. Each point is a polygenic risk score, and the distance to the right is how powerful the score is for predicting that disease. All of the black dots, which you'll notice are the ones furthest to the right, are the ones that we've developed in genomics, and the various coloured dots are those developed by academic groups who specialise typically in one or two diseases, or some of the other companies involved. And to go back to many diseases, I've shown you some of these results before. This compares the difference in risk between someone who has a high polygenic risk score with someone who has a low polygenic risk score for different diseases. So for heart disease, as we saw before, it's about 20-fold. For prostate cancer, it's 40-fold. For breast cancer, it's about 15-fold. For diabetes and atrial fibrillation, it's about 30-fold. Interestingly, for Alzheimer's, it's almost 60-fold. It's also very high for a number of the autoimmune diseases. We don't currently offer a test for Alzheimer's disease because we focus on conditions where there's something that either individuals or their doctors can do to reduce risk. That may well change with some of the drugs that are being developed now, but it's not currently the case. So I've focused so far on results in European ancestry. These scores tend to perform more powerfully when you apply them to individuals from the same ancestry group as most of the training data. And for regrettable reasons, most of the early human genetics data that we studied was from people with European ancestry. That's now being corrected in the field, and it's a really important and very good thing. Within our company, we've developed methods which get as much as possible out of the smaller data sets available for other ancestries. And the figure on the right gives a sense of this. Don't worry about the details, but each row here is a disease. There are different colored dots. The blue dot is the predictive power for individuals of European ancestry. The green dot is for South Asian ancestry, the orange dot for East Asian ancestry, and the purple dot for African ancestry. Now, in UK Biobank, which is where we assess this, we didn't have enough data to test performance for all ancestries for all diseases, which is why there are some dots for some but not other diseases. And the little encapsulated figure on the right summarizes it. It turns out that for people of East Asian or South Asian ancestry, performance is reduced by about 10% compared to European ancestry. It's substantially more for people of African ancestry, maybe 25 to 30%. That's something as a field we must address, and indeed we are. But I would make the point, and it's a really important point, that in all ancestries, these scores are clinically useful now. We should decrease the differences between ancestries, but they're still useful. And indeed, this slide shows some work we've done to do that in a collaboration with scientists in Taiwan. We were able to get additional training data, and this shows how it improved our scores. For each row where they're both there, if you compare so the purple dot is the performance after we'd also trained on the data from Taiwan. And remember, the green dot is what the performance was before we trained on Taiwan. So you can see the difference between the purple dot and the green dot in many cases. So the additional training data improved performance. And if you compare the purple dot with the black dot is performance in European ancestry individuals. For many diseases, they're now the same. For some, performance in East Asians is indeed slightly better. So that gap, which was about 10%, is now much smaller. And that's what we need to do more of as a field, and we're working very hard on finding those data sets to allow us to improve the methods. Okay, so I've talked about a technology which allows personalized assessment of risk for all the common diseases. You only need to do the genetic test once in your life to get the data from which we can calculate these polygenic risk scores for many, many diseases. And that genetic data doesn't change across your lifestyle. That's why you only need to get it once. Sorry, across your lifetime. That's why you only need to get it once. If we think about one disease, like heart disease, most of us won't be in the top 5% of the polygenic risk score. In fact, 5% of us will be in the top 5% of polygenic risk score. But actually, if you think of 10 or 20 diseases, for each of us, there'll be a few diseases where we are at high risk. At the moment, we have no idea what those diseases are. Neither do our doctors or our healthcare professionals. So one way of thinking about this as an individual is we're all going to be at high risk of a few diseases because of the randomness of our genetics. We can find out what they are, and then we can learn what we can do as individuals to reduce that risk and what our health systems can do to reduce the risk. And from a health system point of view, for all the diseases we look at, those systems run screening programs or prevention programs or treatment programs. They need to determine who gets into those programs, and they currently do that with very, very unsophisticated, rather clunky, I might say, measures of risk. So in the UK, all women are offered screening for breast cancer when they turn 50. It doesn't make sense to offer it to all women, because for women who are young enough, their risk of breast cancer is very small. You have to decide whom to offer the mammograms to, and 50 is just used currently as a surrogate for a certain level of risk. But you've seen the figure, there's massive variation between different women in their level of risk. Some women reach the level of risk of a typical 50-year-old when they're 40. At the moment, they're invisible to the system, and they potter through their 40s not being checked. That makes no sense. We should identify those women, and we should target our screening based on risk for individuals rather than just age. There are many other settings where health systems do the same thing. This allows them to do it better. We collaborated with the National Health Service in the UK to run a clinical trial. The clinical trial was about adding polygenic risk to the current clinical risk calculator used for cardiovascular disease, heart disease and stroke. The figure on the right is the data from all 832 participants in the trial. So Q-risk is the name of the clinical risk calculator. So the y-axis plots an individual's Q-risk score. So the x-axis plots an individual's Q-risk score, and the y-axis plots their score after we've added in genetics. As I said, for younger men, genetics is about half the risk. And there's a paper from the University of Leicester recently analysing real-world evidence that shows that when you add polygenic risk into Q-risk, you increase the sensitivity of Q-risk by about 50%. So it makes a big difference. One of the things we wanted to check, and it was the primary endpoint of the study, was whether this fitted in with standard GP, so primary care workflows. And it did. Next question is, how did the patients find it? People were worried about whether they get confused, whether they start worrying about their health. They loved it. 98.5% of them said they found it helpful. In about a quarter of cases, there was a clinically significant change in the patient's risk. And in 13% of cases, the GPs, the doctors, changed their management of the patients. Now, this is a relatively easy and straightforward use case because at the moment, at least in the UK, the doctor has a conversation with the patient that goes along the lines of the following. Well, Helen, for example, we've run an algorithm that combines your age, your sex, your blood pressure, your BMI, your cholesterol level, and so on. And as a consequence of that, we've estimated your risk for cardiovascular disease, heart disease or stroke over the next 10 years to be, I don't know what it is, but let's say it's 12%. And the doctor would say, they'd explain what the risk means, and then say, that's a little bit higher than we'd like. Here are some things you could do, and you might consider going on statins to reduce your risk. When there's genetics involved, the doctor has almost an identical conversation. They now say, well, Helen, we've run an algorithm which combines your age, your sex, your blood pressure, your BMI, your cholesterol level, and an overall summary of your genetic predisposition for cardiovascular disease, and your risk is 18%, and so on. So for the doctors, they're already explaining risk, they're used to doing that, and they're used to explaining it in ways that are accessible to patients. What does the data show? Well, if risk didn't change, every one of these data points would be on the diagonal line. And those that stray for the line are those where, in this half, genetics has reduced someone's overall risk, and here where it's increased them. There are some individuals, about 5%, who went from being, in the UK, the guidelines say that if someone's risk is above 10%, they should be offered statins. There are about 5% of individuals who are over that level when you include genetics, who were not high risk before that. But the groups that the doctors found most interesting were these sorts of individuals who were already high risk, but they became very high risk when their genetics was taken into account. And that they reported, it completely changed the conversation the GP had with the patient and the patient's attitude, both to making changes in their lifestyle and to statins. In the case of cardiovascular disease, we have a very good method for reducing people's risk, statins, which reduce cholesterol. I used to be asked whether, for this special group, because of their genetics, how did we know statins worked? Well, we know because people have looked back through many of the statin trials to see what is the impact of statins on this group with high genetic risk. And it's a win-win. It turns out not only do statins work on that group, they're more effective therapeutically than they are on average. And for those on the medical side, it's interesting. They don't change the amount by which someone's cholesterol is lowered. But this high polygenic risk group has the property that lowering cholesterol by a certain amount has more of an impact on their risk of cardiovascular events than that lowering does on average. So it's a hypothesis, but we think of high polygenic risk people for heart disease as people who have sensitive vasculature, vasculature that's more prone to being affected by cholesterol, and hence lowering it is impactful. The data at the top is from a real-world study from Neil Rish's group. This is the reduction in numbers of events for people on statins, depending on whether they were low polygenic risk score, intermediate or high, and smaller numbers mean a higher reduction, so you can see the larger impact for the high polygenic risk score individuals. This is analysis of a number of statin trials and then trials of the next generation of cholesterol-lowering therapies, so-called PCSK9 inhibitors. Some of the statin trials are primary prevention, so preventing heart attacks and strokes in people who have never had one. Some are secondary prevention, preventing them in people who have already had them. And what's shown is here the absolute risk reduction in people with low polygenic risk score and high polygenic risk score. And you can see a much larger absolute risk reduction. Medical statisticians often like talking about something called the number needed to treat. So the idea is, how many people do you have to treat with statins to prevent one event? And on average, that number is about 50. In the high polygenic risk group, the number needed to treat is 12. Two things that are, again, a technical point, there are two things that are happening there. They have higher absolute risk because they're more likely to have events, and the relative impact of statins is larger, and they both work in their favor. So we can identify a group of individuals who are at high risk. They're currently invisible to the system, and the statins are even more effective there than they are in the general population. We should just get going and start doing it. This work has attracted a lot of attention in the UK as we've gone on various stages of our journey. To give you a sense of impact, in the UK as a whole, for people aged between 40 and 69, there are 2 million individuals who are at high risk for heart disease or stroke who are currently invisible to the system. It's a lot of people. If we identified those and treated them with statins, we'd prevent over 40,000 acute events over the next 10 years. That's a big impact, and estimates of the cost saving to the NHS are about £150 million. That's the cost savings to the NHS. There's much more attention now on the impact on the UK economy through loss of productivity. That's amplified, it's even larger. For breast cancer, there are a large number of women in the UK who are at high risk or moderate risk, as defined by NICE, the clinical standards body, who should, according to the guidelines, be offered mammograms every year. They're currently invisible to the system. If we could detect them and give them the mammograms, each mammogram will be twice as likely to find a cancer. So from the health system's point of view, you're using your prevention tools more efficiently and more effectively. If you use this technology with bowel cancer screening, in order to determine, in the UK there's a two-stage process. There are at-home fecal tests, FIT tests, and people at a high enough level are then offered a colonoscopy. If you use polygenic risk scores in the right way, you would improve the detection rate of colonoscopies by over 20%. Osteoporosis, so bone fragility, typically more common in women, is another area where genetics is really important. If you use this technology in the right way, you would use fewer DEXA scans, but identify more than twice as many people who are genuinely at high risk. And again, there are therapies that can be used for those. In the UK, there's a very large and exciting program called Our Future Health. It aims to recruit 5 million people over 5 years. Part of the offer for those individuals is that they will get information about their genetic risk of disease. We are privileged to be the exclusive partner of the Our Future Health program. They will be getting our risk scores back. So through this program, within a few years in the UK, there'll be millions of individuals who will know about their genetic risk of disease. That was already exciting. Then yesterday, as I said, the government announced its 10-year plan for the NHS. First note, front cover, double helix. That's indicative and, of course, encouraging. And in the 10-year plan, the government promised what they called the new Genomics Population Health Service, available to all by the end of the decade. They were going to implement newborn genomic testing, but also population-based polygenic risk scoring in exactly the ways I've described it, starting with cardiovascular disease. So we have a fantastic opportunity in the UK to lead the world, as we have in a number of other areas of genomics, by implementing these technologies at scale to make a real difference. Another really positive thing about these technologies is they are in effect blind to health inequalities. Health inequalities are a major problem in many countries, certainly in the UK. But if you can get genetic risk for the individuals in the communities that aren't currently well served by the health system, we have the possibility of getting the right tools and the right care to them at the right time. In the UK, we have a product called Health Insights, which is MHRA registered and now available. It's offered by Bupa, which is a large private health insurer, by Spire Healthcare, who run the largest private hospital network in the UK, as part of health checks they offer to individuals, and a number of private clinics, one of which is called WellLife. The current UK test provides reports to doctors, just like in the trial I described, for cardiovascular disease and diabetes. The doctors get an integrated risk score, where the genetic component of risk is combined with their clinical risk tools to get a more powerful sense of risk for their patients. For breast cancer for women and for prostate cancer for men, they learn about lifetime risk. And again, certainly in the case of Bupa, they can direct screening resources in the right way. It's great. Well, it's great for us as a company. It's one of the things we're very keen on is getting these tests and results into the hands of as many people as possible. This can power a whole new wave of prevention, and we wanted to do so. So it's great that it's being picked up by two, and we think by the end of the year by most of the private providers in the UK. But it's really critical in the UK that the NHS doesn't fall too far behind. We also have a test available in the US for a different number of diseases. It's physician ordered, so CLIA certified. It provides reports directly to individuals and to healthcare providers. That's already been offered to large numbers of people in the US and is now recommended by many of the leading healthcare systems, Cleveland Clinic and Stanford among them. So I focused on prevention, and that was indeed the story I wanted to tell about how we can finally use genetics and sophisticated algorithms to really personalize medicine, to help people understand their risks. But that'll just be the first wave of genetics in healthcare. There are others in terms of liquid biopsy that Professor Lowe and his colleagues have pioneered and have already had a massive impact. But there are other changes that will happen in the next 5 or 10 years as we start to have the genetic information available. I've already explained that an individual's response to statins is affected by their polygenic risk score. That will be true for other medications. At the moment, when doctors are prescribing medication, they'll often have a choice. Some medications work for some patients and not others. We don't really know how to distinguish them in advance. Genetics will be a part of that story. When we get the right data, we'll be able to develop scores like these polygenic risk scores, which can guide clinicians in prescribing. They'll also be useful for clinical decision support when doctors have to make choices between radiotherapy and surgery for prostate cancer. At the moment, that's a fairly finely balanced choice, and I understand the answer you get depends on whether you ask a radiologist or a surgeon. But in time, we'll learn that genetics can be part of tipping that balance, and we'll be able to give the doctors that information. In 10 or 15 years' time, I think genetics will be routine in healthcare systems. It'll be routine by then to use it in the ways I've described for prevention, but also by then for many other uses. As I said, some of us have been talking about personalised medicine for 20 years. We've had a centre in Oxford for much of that time. Personalised medicine is now ready for prime time, and it's very exciting. Thank you very much.